Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Do you know over 120 million Americans are dealing with either diabetes or prediabetes? It's a staggering number, and it really makes you think about how this widespread condition could be affecting other aspects of our health.
Absolutely. And one area where the connection is becoming increasingly clear is in cancer.
That's right. And this deep dive is going to take us into some fascinating new research that explores the link between obesity-driven diabetes and a particularly aggressive type of breast cancer.
We're talking about triple-negative breast cancer, or TNBC.
Exactly. You shared a health news article and a scientific paper on this topic, and I have to say, it's pretty eye-opening stuff.
Yeah, the research really delves into why people facing both of these conditions, diabetes and TNBC, often have more difficult outcomes. And it suggests some potential avenues for how we might think about treating this challenging disease.
So let's break down the science and see what we can learn. What exactly did this research look at?
Well, the study was conducted by scientists at Boston University, and they were really trying to figure out why TNBC patients who also have obesity-driven diabetes tend to have worse outcomes.
Like a higher risk of the cancer spreading or metastasis?
Exactly. And the thing is, current oncology practice doesn't really differentiate treatment much for these patients.
So they're not necessarily getting more aggressive treatment even though they may be at higher risk.
Right. And this study wanted to understand why that might be happening at a biological level.
So what was their main theory?
Their hypothesis focused on these tiny vesicles called exosomes, which are released by fat cells.
Exosomes. Okay, do you mind me? What are exosomes again?
They're like these little packages or messengers that cells use to communicate with each other.
Ah, got it.
And in this case, the researchers thought that the exosomes being released by fat cells in a diabetic state, where they're insulin resistant, might be carrying signals that could actually make the TNBC more aggressive.
Wow, so the fat cells are actually sending out messages that could be changing the behavior of the cancer cells. That's pretty wild.
That's exactly what they were looking at.
So how did they study this? Did they do this in people or in the lab?
They started with lab experiments, which is often how these investigations begin. What they did was collect exosomes from mature fat cells, some of which were insulin sensitive, meaning they responded to insulin normally, and some that were made to be insulin resistant to mimic the diabetic state.
Okay, so they had their normal fat cells and their diabetic fat cells. Then what?
They took those exosomes and added them to a type of mouse cancer cell called 4T1 cells, which are a model for TNBC. They're often used in research because they behave a lot like human TNBC cells.
I see. So they're trying to recreate what might be happening in a person with diabetes and TNBC, but in a controlled lab setting.
Precisely. And they watched these cells for 72 hours to see what would happen.
72 hours? That's a pretty short amount of time. But I guess with cells, things can change quickly.
They do. And they were looking for specific changes in the cancer cells, especially something called epithelial-to-mesenchymal transition.
Epithelial to, wow. That's a mouthful. What on earth does that mean?
It's a process where cells change their characteristics. They essentially go from being stationary and well-behaved to becoming more mobile and invasive. And that's one of the hallmarks of metastasis, of cancer spreading.
Oh, I see. So the cells are basically breaking free from the original tumor and getting ready to travel to other parts of the body?
Exactly. And they also tracked how much the cells were multiplying and how many new blood vessels they were forming, which is called angiogenesis.
Because tumors need a good blood supply to grow, right?
Absolutely. And what they found was that the cancer cells that were exposed to the exosomes from the insulin-resistant fat cells showed more of that epithelial-to-mesenchymal transition. They were also multiplying faster and forming more blood vessels than the control group or the group exposed to the exosomes from the insulin-sensitive fat cells.
So even just being exposed to those messages from the diabetic fat cells seemed to be making the cancer cells more aggressive.
It seemed that way. And to further confirm that, they did some tests to directly measure how mobile the cancer cells were. And again, the cells treated with the insulin-resistant exosomes showed a much higher ability to move.
Wow. So that's all in a petri dish though, right? What about in an actual living organism?
That was their next step. They moved from these in vitro experiments, meaning in the lab, to an in vivo model, which means using animals. In this case, they used mice.
Okay, so how did they do that?
They took their 4T1 mouse cancer cells and treated them with the different types of exosomes again for 72 hours. Then they injected these treated cancer cells into the mammary fat pads of the mice.
So they're basically trying to see if what they observed in the lab dishes would also happen in a more realistic setting.
Yeah, and after about 2 weeks, they examined the tumors that had grown in the mice, as well as other organs like the lungs and the brain, to see if the cancer had spread.
And did they find that it had spread more in the mice that received the cells treated with those insulin-resistant exosomes?
Yes, the tumors in those mice had higher levels of the proteins associated with that epithelial-to-mesenchymal transition, as well as markers of cell proliferation and blood vessel formation. So it was like a replay of what they had seen in the lab, but now in a living animal.
And what about the brain specifically? Did they see more spread to the brain in those mice?
Yes, that was a very important part of their findings. They used a special technique to grow and study the cancer cells that had spread to the brain. And the number of those metastatic colonies in the brain was significantly higher in the group that got the cells treated with the insulin-resistant exosomes.
So that's a pretty big deal, right? Because brain metastasis is often very difficult to treat.
Absolutely. And it wasn't just the number of those cells. The researchers also noted that the shape of the cancer cells that had spread to the brain in that group was different. They had more of those mesenchymal-like features, meaning they looked more like they had gone through that transition to become more mobile and invasive.
So it seems like those messages from the diabetic fat cells were not only making the cancer cells more aggressive, but also maybe even helping them get into the brain more easily.
That's one of the key takeaways. And the next question, of course, was why? What was it about those exosomes from the insulin-resistant fat cells that was having this effect?
Right, so they needed to dig even deeper into the biology.
Exactly. They started analyzing the contents of those exosomes, and they found a specific molecule called a microRNA, or mRNA.
Okay, microRNA. I think I've heard of that. What exactly is a microRNA?
It's a tiny piece of genetic material that can actually regulate the activity of other genes. It's like a dimmer switch that can turn the volume up or down on certain genes.
Got it. So they found a specific microRNA that was more abundant in the exosomes from the diabetic fat cells.
Exactly. It was called miR-145A3P. And they found that it had sequences that could potentially bind to and control genes involved in something called mesenchymal to epithelial transition, which is basically the reverse of that epithelial to mesenchymal transition we talked about earlier.
Ah, okay. So it could potentially be turning down the volume on the genes that help the cancer cells become less mobile and invasive?
Right. And it could also be regulating genes involved in tumor suppression, which are the genes that normally help to keep cancer in check.
Oh, wow. So this tiny little molecule could be having a huge impact on the cancer's ability to grow and spread.
That's what they're suggesting. And to really nail this down, they directly treated TNBC cells with just that microRNA, and it did indeed increase their ability to migrate.
So it's like they isolated that one signal from the fat cells and ensured that it was enough to change the behavior of the cancer cells.
Right. And to tie it all back to what we see in patients, they then analyzed data from actual breast cancer patients.
Oh, so they weren't just working in the lab and with mice, they looked at human data too.
They did. And what they found was that the patterns of those microRNAs they identified in their lab and animal models were actually linked to how well patients survived.
So if patients had higher levels of that specific microRNA, were they more likely to have worse outcomes?
Yes, their findings supported that connection.
That really strengthens the case that this might be an important mechanism in real people.
Right. It's still early research, but it definitely adds to the growing body of evidence that our metabolic health can have a significant impact on cancer.
So let's step back for a minute and summarize what we've learned. What are the main takeaways from this study?
The big picture here is that this research suggests that insulin resistance in fat tissue, which is a hallmark of diabetes, changes the environment around breast tumors in a way that makes TNBC more aggressive and more likely to spread.
And specifically to the brain, which is particularly concerning.
Exactly. And it does this through these exosomes, those tiny messengers that carry signals, including this specific microRNA, miR-145A3P, that can make the cancer cells more mobile, more invasive, and better at establishing themselves in other parts of the body.
So essentially, it's like the diabetic fat cells are sending out messages that tell the cancer cells to go, grow, and spread.
That's a good way to put it.
And the implications of this for treatment could be huge.
They could be. Because as we mentioned, currently, doctors don't typically factor in whether a patient has diabetes when deciding on a treatment plan for TNBC.
Right. But this research suggests that maybe they should be.
Yeah. It highlights the need for what they call special consideration for these patients. Maybe they need closer monitoring for metastasis or maybe even different treatment approaches that address the metabolic aspects of their condition as well.
And given the global rise in obesity and diabetes, this is an issue that could affect a huge number of people.
Absolutely. The researchers emphasize that point. This isn't just some rare phenomenon. It's potentially a global health concern.
And it's yet another reminder that cancer doesn't exist in isolation. It's influenced by everything else going on in our bodies.
That's a really crucial point. Our overall health, including our metabolic health, plays a big role in how cancer develops and progresses.
Of course, no scientific study is perfect. Were there any limitations to this research that we should be aware of?
Well, a lot of it relied on a mouse model of TNBC, so we always have to be a little cautious about directly applying those findings to humans.
Right. Mice aren't people.
Exactly. But they can give us valuable clues. And the researchers did look at human patient data as well, which is encouraging.
And I guess metastasis is a really complex process, so even if they've identified one key mechanism, there are probably many other factors involved.
That's true. And it's also worth noting that in their animal experiments, they did see an increase in lung metastasis, but it wasn't statistically significant.
Meaning it could have been due to chance?
Possibly. They suggested it might be because the specific type of mouse cancer cell they used is already very prone to spreading to the lungs, so it might have been hard to detect a further increase.
I see. So what did the researchers have to say about their findings?
Well, the lead researcher, Dr. Gerald V. Dennis, was very enthusiastic about the potential of this work. He said he hopes this research will lead to better and more personalized therapies for patients with aggressive breast cancers, especially those with metabolic disorders.
So he sees this as a real step forward in understanding how to treat these cancers more effectively.
Exactly. And he also emphasized the importance of not just treating the cancer itself, but also addressing any underlying conditions like diabetes.
It really is about taking a more holistic approach to health, isn't it?
It really is.
So what do you think? Where do we go from here?
I think this research raises a lot of important questions for both scientists and clinicians. We need more research, of course, to confirm these findings in humans and to really understand all the nuances of this connection between diabetes and TNBC.
And to figure out how we can use this knowledge to improve treatment strategies.
Exactly. But it also raises questions for all of us about how we can take better care of our metabolic health, not just for the sake of preventing diabetes, but potentially for reducing our risk of developing aggressive cancers like TNBC.
Absolutely. For anyone who's interested in learning more about this study, we'll be sure to include links to both the news article and the scientific paper in the show notes.
It's definitely worth a read. It's fascinating stuff.
And it really leaves us with a lot to think about. Could taking a more holistic approach to cancer treatment, one that really considers the whole person and their metabolic health, be the key to making real progress against these aggressive cancers? It's certainly something to ponder.
I think so. It's clear that there's a lot more to learn about the connection between our metabolism and cancer, and this research is a really exciting step in that direction.
Thanks for diving into this with me. It's been a really thought-provoking discussion.
My pleasure. Always happy to talk about this important work.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.